Hello everyone, my name is Nirmala Chinnapareddy. I am a scientist and study director at Outer Sciences Preclinical Seattle. Thank you so much for your interest in my poster that will be presented at the 45th Annual Meeting, American College of Toxicology 2024 in Austin, Texas. I will give you a brief review of the overall summary of different immunomodulatory approaches in preclinical gene therapy studies. Immunomodulatory drugs are increasingly significant in gene therapy because they help modulate the immune response to therapeutic transgenes or vectors. They do this by either minimizing immune reactions to vectors, or by preventing transgene immunogenicity, or by modulating inflammation at target sites, or either by improving tolerance to gene editing techniques. Immunomodulatory drugs play an essential role in enabling safer and more effective gene therapy by managing potential immune responses that can compromise the treatment success. Specific immunomodulatory drugs used in the gene therapy include corticosteroids, that could be example prednisone, dexamethasone. These are widely used to suppress inflammation and immune responses to gene therapy vectors and transgene proteins. And also there are another set of immunomodulatory drugs. They are called calcineurin inhibitors. They include tacrolimus and cyclosporine. These are used for more targeted suppression of immune responses, especially in conditions requiring long-term immunomodulations. And there are another group of immunomodulatory drugs. They are called mTOR inhibitors, for example, rapamycin. And these drugs help modulate T cell activation and proliferation, and these are beneficial in reducing immune responses to both the vectors and the transgene. And there are other last class of drugs called monoclonal antibodies, for example, rituximab. These kind of drugs are used to deplete specific immune cell populations like beta cells, which can either reduce the formation of anti-drug antibodies. The objective of this study or the poster was to investigate the commonly used immunomodulatory drugs or pre-medications in gene therapy studies. We did a survey by screening the data from 30 gene therapy studies that were conducted at our testing facility that utilize adeno-associated virus, AAV vectors, or lipid nanoparticles or LNPs over the past 3 years. And out of these 30 gene therapy studies that were screened, only 15 studies used the immunomodulatory drugs. We further screened those only 15 studies to look for either frequency or the timing or the route of administration or how long those pre-medications were used. And also we looked either they were used in single drug or in combination. And also we looked for clinical observations, and we also looked for biomarkers such as cytokines and complements and clinical pathology evaluations. Among these studies reviewed, like I said, 15 out of 30 utilized the pretreatments or immunomodulatory drugs, with dexamethasone and prednisolone being the most commonly used, followed by various combinations of other treatments. Dexamethasone was used like 2 milligram per kg IV bolus. It was mostly used 1 hour prior to dosing, and out of 15 studies, 6 of them used dexamethasone alone. And 3 studies used prednisolone at the dose of 1 milligram per kg, and they were all used orally, and they were given daily during the dosing period. For the entire dosing period, they were administered every day. And diphenhydramine was also given at 2 milligram per kg intramuscularly, and it was given like 15 to 30 minutes prior to dosing. And all these remaining studies, like there was only one study for each pre-medication or the combination of pre-medications. And there was another study with prednisolone and diphenhydramine combination. Prednisolone was used 3 milligram per kg, and diphenhydramine was used 5 milligram per kg, and prednisolone was used orally daily from day -1 to day -1, and 30 minutes pre-dose it was given. And the diphenhydramine was given intramuscularly. Both were given pre-dose. There was one study where tocilizumab was given at 8 milligram per kg body weight IV infusion only on day -1. And there was one study with the tocilizumab at 8 milligram per kg body weight IV infusion on day -3. And the same study also was given with the tacrolimus at 0.05 milligram per kg intramuscular daily from day -1. There was one study with rituximab at 750 milligram per meter square IV infusion on days -15 and 1. And same study had diphenhydramine 4 milligram per kg body weight IM, days -15 and 1, and sirolimus 2 milligram per meter square per day orally daily from day -3. And the one last study that we had in survey had a sponsor's own immunosuppressants. They were like two immunosuppressants, either immunosuppressant 1 or 2. The second immunosuppressant had famotidine as well. The immunosuppressant 1 was given at 50 milligram per kg or zero 0.1 or zero 0.5 milligram per kg, and the famotidine was given at 0.7 milligram per kg. All were given IV bolus, and either they were given on day -7, -3, or on day 1 pre-dose, and thereafter once weekly during the entire dosing period. Okay, when we were doing the survey, we also looked for the clinical symptoms or observations. Out of 15 studies surveyed, all 14 studies had no abnormal clinical symptoms related to immunomodulatory drug administration, while only one study had some abnormal clinical symptoms that were ruled out as due to the exaggerated pharmacological effects of tesiracine. The most common clinical observations in animals administered with the immunomodulatory drugs were consistent with those seen in control animals and typical for animals maintained under similar laboratory conditions. And these observations included like bruising, abnormal skin coloration, changes in feces, hair loss, abrasions, scab, or crust. And we also surveyed these studies for the types of biomarkers they were screened for and types of changes that happened. So, among 15 studies surveyed, only 6 studies evaluated cytokines. Of these 6 studies, most of them showed no changes in cytokine levels, while only one study reported an increase in cytokine levels. And out of these 15 studies we surveyed, 5 studies evaluated complement levels, and of these, only 2 studies showed no changes in complement levels, while 3 studies reported an increase in either one or more measured complements. And these biomarker changes, like cytokine and complement, were mostly seen as transient changes, like increased levels, either in MCP1, SC5B9 complex, or BB-RC3A complement fragments. And when we surveyed the clinical pathology parameters or findings in these animals administered with immunomodulatory drugs, again, out of these 15 studies surveyed, mostly all studies had clinical pathology parameters evaluated. These clinical pathology findings included various levels of decreased lymphocytes, monocytes, neutrophils, eosinophils, or platelet counts, and also there were decreased ratios of albumin to globulin ratios, or increased fibrinogen, a prolonged prothrombin time, or reversibly increased liver enzymes such as ALT and AST, and the total bilirubin and our creatinine kinase levels. Overall, these biomarker and clinical pathology findings, when reported, were either minimal or mild, and mostly they were transient and reversible, and were evidence comparatively less in studies that used dexamethasone as a pretreatment. Overall, in conclusions, immunomodulated drugs are essential for minimizing immune reactions to therapeutic transgenes and vectors, and they help reduce the transgene immunogenicity, modulate inflammation at target sites, and enhance tolerance to gene editing techniques. The data we reviewed in this presentation indicate that corticosteroids such as dexamethasone and prednisolone are most commonly used immunomodulatory drugs in gene therapy. Either they could be used as standalone treatment or in combination with other premedications. Among these studies we screened, dexamethasone was most popular and had a greater suitability as a pretreatment option.